Our third speaker is Honi from MIT, and he's going to talk about animal-to-animal variability in hippocampal remapping.
Yes, thank you. Nice to talk to you all. So, yeah, I'm going to be talking about animal-to-animal variability in hippocampal remapping. So, briefly, hippocampal remapping, there are place cells in the hippocampus. Each place cell corresponds to a particular location in an environment. Fires particularly when the animal's at a particular location, and when you move to another environment, the place fields will either move or stop firing altogether. And this is called hippocampal remapping because the idea is that there's different maps for different environments. So, you know, the standard way to say this is different room, different map, same map. But recent theory work that I had done discusses how animals actually have to infer environment identity, and they're not actually given directly labels for which environment is which makes it a much more complicated process than you might have thought. And in particular, we had suggested that this might be an explanation for animal-to-animal variability, which had been mentioned in the literature but never previously shown. So what we wanted to do is reanalyze previously collected data in order to demonstrate the existence of animal-to-animal variability. So the data that we were analyzing is from this paper in PNAS from the Moser lab. They recorded the animal it was the name of the paper is 11 maps for 11 rooms. And basically they trained the animal in a familiar environment, showed the animals 5 novel environments on the first day, and then repeated the first novel environment and repeated the familiar environment. Similarly on the second day, 5 novel rooms, repeated the first novel room from that day, and then repeated the familiar room. And what this protocol allows them and us to do is to compare both in between different environments and for repetitions of the same environment. So here's a single place cell. This is a particularly active place cell, but this is a single place cell in all of these different rooms. You know, nicely you can see that for repetitions of the familiar room, the place field is in the same location, which is what you would expect if the map was the same for repetitions of the same environment. And when you go to a different environment, the place field is in a different location, again, as you would expect if there's different maps for different rooms. And also you have for these immediate repetitions, the same place field in the same environment. And even for these distant repetition, where novel room 1 is repeated at the end of the day, the place field is also the same. So again, this is a very clean cell, very nice, but just to demonstrate, you know, the idea behind remapping. So we can calculate a score called a rate map correlation between different sessions. So for example, if we compare, say, this N2, the first half and the second half, the rate map correlation for that is 0.94. And the rate map correlation for this session versus this session is 0.14. So you'd expect, you know, correlations near 0, the place fields are different. Correlations near 1, the place fields are the same. So we can average these over all cells to see what the average rate map correlation for a particular pair of sessions is. So what we can see, let's focus on this side. So this is the average rate map correlations for repetitions of the familiar room. And you can see there's a wide variability in the amount of remapping that occurs. But what's interesting is that these are colored by animal. And you can see this yellow animal has consistently high correlations. This blue animal and the purple animal have more consistently low correlations. What's interesting is if you look at repetitions of the novel rooms, you also see variability, you also see clustering by animal, and you also see that the animals that have consistently high correlations for repetitions of the familiar also have high correlations for repetitions of the novel. So we can quantify this in a variety of different possible comparisons for, you know, these sessions. How similar are they in these repeated sessions, and how similar are they in this other set of repeated sessions? And you can see there's a high correlation for all of these different comparisons, meaning that animals that have strong correlations in one set of environments also have strong correlations in another set of environments, and animals that have low correlations in one set of environments also have low correlations in another set of environments. And then what that implies is that animals that have that the maps repeat very strongly for repetitions of environments are consistent with that, whereas animals that have very weak retention of maps for repetitions of environment are also consistent in that. So to summarize, individual animals have different remapping tendencies. These remapping tendencies are consistent within animals for different types of comparisons. And there was a variety of controls that I did, which I can discuss if you're interested. This work was done by Parsa, who was an undergrad working with me, and then my advisors and funding.
Well, thank you very much, Honi, for the presentation. We have actually a couple of questions, and the question is, do you want to ask them yourselves or do you want me to read them? We have first up Eleanor Duvel. Okay, then I'm going to read this question. The question is, are all animals of the same age, gender, etc.?
That's an excellent question. They're all of the same gender. I assume they're all of the same age, but I don't have that information. But it's an interesting question because previously it had been shown that age can cause differences in tendency to remap versus not remap. So that's a very good suggestion.
Does that answer your question, Eleanor? Yeah, okay, it does. Very good. The second question is from William Mao. William, do you want to ask the question yourself or do you want me to read it? Okay, I'm going to read it. The question is, how much RMC clustering can be explained by the number of neurons recorded?
So that was actually one of the controls that I did. Let me just pull it up. So this red animal is the animal that had the least number of cells, 25 cells. This blue animal had a much larger number of cells. I subsampled 25 cells from the blue animal, and this is the variety of different RMCs that you would get from those subsamples. You know, so it doesn't look like it's due to the number of cells. I did a similar thing here where I just subsampled 25 times from all these different animals, and it seems that the clustering is maintained even with the subsampling.
William, do you want to follow up? Do you want to ask another question, or does it answer your question? Okay, he says, thank you. And then I've got a question here from Jason Moore. Jason, do you want to ask the question yourself, or do you want me to read it? Okay. Are the correlation coefficients bounded to be positive somehow? Shouldn't the correlation between the two maps with a peak in non-overlapping places ought to be negative, or do all of the zero bins negate that effect?
This is a very, very interesting point. So actually, this is the one that I want to show. So they're not bounded to be positive, but it turns out that the rate map correlations are mostly positive. And I think that the reason for this is because of the fact that the place fields are either in the same location or they're in a different location. But since most of the map since most of the environment has zero firing, those are, if the place field moves, then most of the map, most of the environment is still going to have the exact same value for firing rate in most bins. So you can't really get negative correlations very easily, since most of the map is just zero in both conditions. Does that make sense? And I'm not 100% sure that that's the reason, but that's my guess.
Jason, does that answer your question? Thanks. We've run into this to that as well. That's what Jason says. We do have more time. It is we have actually officially 5 more minutes to ask questions. If there are no specific questions with respect to Honi's talk, feel free to ask questions about Kate's and Guangwei's presentations. Or if the... Yeah.
Oh, okay. I have a question for Honi. So I think because this like place cells always attract my attention, because I was wondering like how the animal gonna use the visual information with the like trying to form internal space about the environment. Because you change the environment sometimes it's mostly regarding with the visual guidance, like, right? So yeah. How do you think they are related with the change of this visual information? And have you ever tried to like map the place cell in complete darkness, and would they still try to remap?
Okay, I'm going to answer the first question and remind me if I missed the second. So yeah, the information that the hippocampus is getting, the visual information, is highly, highly processed. But it's not only visual information that the hippocampus is getting, it's also getting other sources of information. You know, one thing that people talk about a lot is motion information. For example, you know, when the animals move to room, it feels and experiences being moved. And so it knows in a sense that it's in a new room because of that experience of being moved in between. So visual information isn't the only source of information that the animal has. And with respect to like, how does the animal know whether it's different based on the visual information? That's a very, very difficult question. And you get extremely different results depending on the particular protocols. And that's really what I was talking about in the eLife paper, the theory paper that talked about why it is that you get all of these different remapping behaviors for all of these different training protocols. With respect to the darkness, there was a paper, I think by Quirk in the 90s sometime, that actually they turned off the lights and it caused remapping. But it's hard to say whether it was causing remapping because the fact of turning off the light was something significant enough to induce remapping, or because the animal like lost track of where it was. So that, you know, becomes a little bit of a difficult question. Yeah.
Thank you.
I wanted to ask a question to the listeners, if you don't mind, which is, Eleanor had mentioned that age might be playing a factor. Does anybody have any other ideas of what might be playing a role in this?
Eleanor asked a second question in the Q&A, so I will stand in for her for a moment. She also asked, could there be subtle differences in the anatomical locations of the tetrodes, for instance, on the proximal distal axis?
Yes, that's also a, that definitely might be, and there's definitely reason to believe that that might be playing a role. And I also don't have that information. But the one thing that I do find interesting is that the subsampling seems to have a correlation within the animal, suggesting that, which I'm not sure whether that argues for or against that hypothesis, but it suggests that different subsets of cells also have different remapping tendencies, even within an animal. And so on the one hand, you could say that means it's probably not due to anatomical location because these subsamples are being taken uniformly over the different cells that were recorded from that animal. Or you could say that maybe it does play a role because by chance you're getting subsamples that are somewhere along the recording axis. It's an interesting suggestion.
Thank you very much. I'm afraid it is now 4:45, at least here in the UK, and that means we have to, unfortunately, close this room now. However, I would like to thank the speakers for their nice presentations, as well as the audience and the panelists for the lively discussion that we have. And please, for the audience, please get in touch with the speakers in order to answer all the questions that you have. There were some more suggestions there. So then I say goodbye and thank you.